Welcome to our seminars in oncology. It's my distinct pleasure to be able to introduce Anna Sablina. Anna and I go way back to her postdoc days. She did her postdoctoral fellowship initially at the Cleveland Clinic and then Bill Hahn's lab in sort of from 2005 to 2009, where I can honestly say we all looked up to Anna, knew she was going to go on to do great things, published a very nice Cell paper at the time focused on PP2A and RAS regulation, and was recruited to start her own lab at the VIB KU Leuven Center for Cancer Biology in 2010. And since that time, she's risen to the rank of associate professor and has really published a number of very nice papers, including a science paper from several years ago looking at the role of LZTR1 in RAS ubiquitination. And I'm sure we'll hear some of that and some of her new work. So it's really great to invite you back, I wish in person, but for now virtually. Welcome, Anna. And I'm also supposed to tell everybody, please put your questions in the Q&A, not in the chat, and we'll answer them at the end. So yeah, we'll turn over.
Okay, first of all, of course, thank you for the invitation. I'm really happy to present the stuff that we recently published, and of course, quite some unpublished data that we just recently generated. It's not moving somehow. Oh, Claudia, I couldn't.
Yes, are you trying the arrow buttons?
I guess. Okay.
Oh, there you go.
So my lab has a long standing interest in how the RAS GTPase proteins are regulated. And of course, the RAS signaling pathway is quite well known for its role in human disease. It's one of the most frequently activated node in different types of human disease. And specifically, we are interested of how the RAS GTPase is regulated in case of the proteostatic regulation. So, and with a tremendous improvement of mass spectrometry approaches, it's getting quite clear that all RAS family members can undergo ubiquitination at multiple lysins. So, and just a few words about how ubiquitin system is functioning. Because when I'm starting to talk about protein ubiquitination, a lot of people assuming that I'm talking about protein ubiquitination that would lead to proteasomal degradation. But the reality is much more complicated because ubiquitin can form different types of polyubiquitin chains. And while some of them could in fact lead to degradation, many of these chains leads to different physiological, can have different physiological functions. And it could be polyubiquitination, but it also could be just conjugation of one ubiquitin. And monoubiquitination can also have both degradative and non-degradative functions. And we shouldn't forget that ubiquitination is a reversible process. All this type of change could be erased by deubiquitinases, and this could lead to complete deubiquitination of the substrate. So, and if we are going back to RAS GTPases, we believe that the majority of the RAS-like GTPases can undergo mostly mono and deubiquitination. And here is kind of the thing that is quite challenging and the kind of a common question that I'm getting, why we don't normally see this monoubiquitinated protein in our bloods. And the major challenge to detect, there is a detection of monoubiquitinated proteins. Because if you try to pull down monoubiquitinated proteins, it's not so efficient because, of course, polyubiquitin chains have a much higher affinity to anti-ubiquitin resin compared to monoubiquitinated proteins. And another issue is that deubiquitinases are extremely robust enzymes. So it's quite difficult to inhibit the activity of deubiquitinase. And the majority of deubiquitinases, they're based on blocking of active cysteines that are essential for deubiquitinase activity. So the majority of these non-specific deubiquitinase inhibitors, they have quite strong cross-linking activity. So when you try to inhibit its deubiquitinases, like we are doing it for example, for phosphatases, immunoblots are getting really ugly. So the advantage now, because again, mass spectrometry-based approaches are getting better and better. So for majority of our experiments, we are switching from this biochemical purification of ubiquitinated proteins. We are mostly trying to use mass spectrometry-based approaches. So, going back to RAS GTPases, we can clearly see that they can undergo monoubiquitination. But then our question is how this monoubiquitination can modulate and control the activity of RAS proteins. So, and it's could be, we can have quite some options for this. The first things I would think is that, of course, monoubiquitination could affect the interactome of RAS proteins. Because if you're getting this ubiquitination, of course, you can attract quite some protein, especially if you have ubiquitin binding domains. But there are also some indications that ubiquitination can allosterically affect the structure of RAS proteins and then facilitates the interaction. Or another option is if ubiquitination is occurring at the site of the interaction, it could sterically hinder the interaction. So basically, monoubiquitination can dramatically affect the interactome and the interaction landscape of the RAS GTPases. But it's not only the case, because it's quite well known that ubiquitination could also affect the behavior of RAS proteins on a membrane. Because to reach a full activation, RAS proteins should form clusters. But in case of the ubiquitination, this clustering could be affected, and then it could inhibit the activity of RAS proteins. Or another option, if ubiquitination is occurring quite close to C-terminal, it could also affect the interaction of RAS proteins with the plasma membrane. So, and just a bit more specific examples of, in fact, how ubiquitination and different lysins could differentially affect RAS activity. So we did quite some in silico modeling It was done in collaboration with a group of Rus Nusimov from NCI. So we were attaching ubiquitin to different ligands and see how it would affect its interaction and its binding to the plasma membrane. So, and as a first example, we look for conjugation of ubiquitin to lysine 128. Actually, it's one of the most abundant ubiquitination. At least we could detect it in all our mass spectrometry experiments. So, and when we tried, okay, what could be consequences of this ubiquitination, we could clearly see that when we attach ubiquitin to lysine 128, it can create an additional surface to the GTPase activating protein P120 GAP. So suggesting that that ubiquitination at this specific residue could facilitate the interaction with P120 GAP. And we were able to biochemically prove it. We just conjugated attached ubiquitin to RAS using disulfide linkage. We mutated 128 lysine to cysteine, and we did the same for C-terminal glycine of ubiquitin. And we were able to obtain and purify a nice amount of ubiquitinated RAS. So, and then we mix non-ubiquitinated and ubiquitinated RAS with P120 GAP. And we could clearly see that, in fact, when we have conjugation of ubiquitin, it leads to much nicer binding of P120 GAP to RAS. And it's also associated with increased GTPase activity of RAS. So this suggests that this ubiquitination could suppress RAS activity by increasing its GTPase activity. So, and another example is ubiquitination at lysine 170, which is located quite closely to C-terminal part of RAS. So, and you can imagine if RAS is about 20 kilodalton and ubiquitin is about 8 kilodaltons, how it would kind of affect this interaction with the plasma membrane. So we did, again, in silico modeling. So, and in this case, we use fully lipidated form of RAS, farnesylated and palmitoylated. And when we add in this fully lipidated RAS to the in silica to the membrane, we can see that it would immediately attach to the membrane. But it was not the case if we were using RAS that ubiquitinated at lysine 170. In this case, even we were doing this modeling for about 6 months, we couldn't detect any attachment. Couldn't see any attachment of RAS to the membrane. And the reason was that ubiquitin has a hydrophobic patch, and this hydrophobic patch sequesters this lipidated tail of RAS and induce detachment of RAS from the plasma membrane. So again, we were able to confirm it using RAS knocking. We generated ubiquitination deficient arginine mutant of HRAS, and in fact, we see that it leads to increase of membrane fraction and decrease in cytoplasmic fraction. So then it's clearly indicate that ubiquitination not only affects that protein-protein interaction, but it also could affect the interaction of RAS with plasma membrane. And of course, are
you okay, Dr. Sablina? I think she may be frozen. Please just bear with us for a few seconds.
Transatlantic connection.
Yeah, oh boy. We're sorry for this interruption, audience. Please bear with us for a few seconds while we try to get her back. Okay, Dr. Sablina?
sorry.
It's okay. Can you put your presentation back up? Because we seem to have lost that.
Okay.
Just hit share screen at the bottom.
Okay. Okay, sorry.
No worries. It's okay. Yep.
Issue.
Yep. There you go.
So, and as we can clearly see that ubiquitination can affect RAS activity, it would suggest that regulation or dysregulation of ubiquitination could somehow to contribute to human disease. And of course, if there is enzymes that contribute to this ubiquitination, these enzymes could also could be associated with different KRAS dependent KRAS driven diseases. So that's why we decided to look and to search for enzymes that would specifically regulate ubiquitination of the RAS proteins. And for this, we decided to use kind of unconventional approach. We decided to wrap RAS interactors using intact mammalian cells. So what we did, we fused RAS proteins with GAC, and then overexpress them together with FLAG-TAC with VG. So, and when we co-express with VG with HRAS, it induces the formation of kind of viral-like vesicles. And in these vesicles, we were getting trapping of all RAS interactors. So we purify these vesicles and determine their contact by mass spectrometry. So, and one of our top scoring proteins was LZTR1. And we were quite, and at that time, nothing was known about this protein. It even was called the lysine zipper-like transcriptional factor. So it was just, the name was given just by some homology. But there was some indication that this protein has CALG domains and BTB back domains that is quite typical for Kalin-3 ubiquitin ligase complexes. And again, we were quite happy to see that we could find not only LZTR1, but we also could find Kalin-3, suggesting that LZTR1 Kalin-3 complexes could directly control RAS ubiquitination. So to confirm this, we did a reciprocal pull-down experiments. In this case, we use LZTR1 as a bite. And again, we could see that we could pull down both H and KRAS as well as Kalin-3. Again, indicated that LZTR1 can interact with different RAS isoforms. And then we put quite some efforts to confirm the interaction using pull-down experiments. But of course, the goal was to detect interaction of endogenous RAS and endogenous LZTR1. At that time point, there was no good LZTR1 antibodies. That's why we perform endogenous tagging and introduce hybrid tag in terminal part of LZTR1. And using this approach, we could clearly see that, in fact, there is an interaction between pan-RAS and LZTR1. And what is even more importantly, we did unbiased ubiquitinome analysis. We use maps purified from 3 wild-type LZTR1 embryos and 3 LZTR1 knockout embryos. And we could see that LZTR1 knockout completely abolish ubiquitination of RAS, at least at lysine 130. Unfortunately, in these experiments, we were not able to detect NRAS and KRAS. And the reason is that both NRAS and KRAS are more lysine rich. So it's much more difficult to identify them by mass spectrometry because we have to use trypsin for this ubiquitinome analysis. We couldn't switch for any other of digestion enzymes. But even then, biochemically, we could really see that LZTR1 could control ubiquitination, not only HRAS, but also N and KRAS. Indicated that it could be one of the major enzymes that is involved in control of RAS ubiquitination. And of course, the next question was how this ubiquitination could affect the activity of RAS proteins. So, and we can nicely see that if we have loss of LZTR1, we could see a nice increase in RAS activity. So we did it in several systems and in different types of cells, and we are getting quite consistent results. That we can see both an increase of RAS activity, and we can see an increase of the MAP kinase activity. So these results were published, and we and others can quite nicely demonstrate that the LZTR1 Kalin-3 complex can ubiquitinate different RAS isoforms, and it could this ubiquitination could lead to suppression of the MAP kinase pathway. But what is the connection of LZTR1 to human disease? So we were quite lucky that I would say 3-4 years ago, there was really a lot of publications that were genetic studies that identify LZTR1 as a gene mutated in different types of human disease. So the first study was about LZTR1 mutation in glioblastomas. Then there were the studies, cell publications that LZTR1 also associated with hepatocarcinoma. And also germline mutations were identified in schwannoma patients, as well as a germline mutation of LZTR1 were identified in Noonan syndrome patients. And again, it could suggest that there is a direct link between LZTR1 and RAS pathway, because Noonan syndrome, it's a RAS-related disorder. All mutations that are associated with Noonan syndrome somehow related to the RAS signaling pathways. So we decided to look for this mutation and see what could be consequences of mutations that were associated with different types of human disease. So all these mutations that were identified, they are really spread out through the whole gene, with some mutations that are recurrent, but not all of them, but quite some. And just to remind that LZTR1 is an adapter protein. So to be fully functional, it has to be able to interact both with the substrate as well as to interact with Calin-3. So, and then we started to look for different mutation located in different domains and see for their consequences. So first, we will look for mutations that are located in the Kelch domain, which are important for the substrate binding. And what we found that all tested mutations led to inhibition of the interaction between pan-RAS and LZTR1. We also tested mutations that are located in BTB back domains. These domains are responsible for Calin-3 binding. And again, we can see that this mutation leads to decreased ability of LZTR1 to interact with Calin-3. So this suggests that at least the majority of these mutations are loss of function. And there was one exception. It was a mutation at 812, proline mutation. This mutant was able to nicely interact with both RAS and Calin-3. But to our surprise, it still was inactive because we do not see any activity of this mutant in case of its ability to mediate ubiquitination of RAS. So we started to look more closely to this contradiction, and this brought us to another Calin-3 substrate adapter that is SPOP1. SPOP1 is also a quite known tumor suppressor gene, and it's amazing because it has a BTB domain that is really similar to what we have in case of LZTR1. So there's a really high level of homology between these two proteins. And the BTB back domains of SPOP1, they're essential for dimerization and the formation of membraneless organelles of SPOP1 that are sucking kind of the whole SPOP1, Calin-3 and its substrate. So basically SPOP1 has a behavior that is typical for liquid phase separated compartments. So that's why we kind of started to see whether it could be also the case for LZTR1. So we checked whether these 812 mutants could form dimers, and what we can clearly see is that this mutation abolishes the ability of LZTR1 to form dimers. And what we could also see that wild type LZTR1 can nicely form this puncti, this membranous organelles. When we overexpress this 812 mutant, we could see only diffuse cytoplasmic localization of LZTR1. This suggests that, in fact, a specific mutation could abolish the activity of LZTR1 by disruption of this membranous organelles. And it also suggests that LZTR1, for kind of full activity of LZTR1, you need this liquid-liquid phase separation fraction to be fully functional. And to really form these organelles, you need a certain concentration of LZTR1. Because otherwise, the complex, this organelles has not formed, and it could not perform its ubiquitin ligase activity. So we decided to test it. And to test it, we endogenously tagged LZTR1 by GFP. And what we can see, if we were able, when we were able to find clones where both alleles of LZTR1 attacked with GFP, we could clearly see the formation of this puncture. But when we induce deletion of one allele, this puncture disappeared. So this suggests that, in fact, to be fully active, you need a certain concentration of LZTR1. So, and why it could be important in case of human disease. So if we look to TCGA data, we can clearly see that LZTR1 loss of heterozygosity is observed in about in a wide range of human cancer. So this suggests that even loss of one allele could fully block LZTR1 activity, and that it means that LZTR1 potentially could be GAPA insufficient tumor suppressor gene. So what we have that LZTR1 loss of function could be associated with a wide range of human disease. And we can clearly see it at the level of biochemistry and in numerous genetic studies. But of course, we would like to model and to see ways of it confirm this using in vivo models. So we started to generate LZTR1 knockouts and see how these knockouts recapitulate different human phenotypes. So first, we generated full body LZTR1 knockout. Unfortunately, full loss of LZTR1 was embryonically lethal. So these embryos were dying around E13.5 due to massive bleeding. But fortunately for us, even loss of one allele could lead to quite some phenotypes, and these phenotypes are nicely overlap what is observed in Noonan syndrome patients. So you can clearly see that LZTR1 heterozygous mass is smaller. It also has quite some bone defects. The eyes in this mice are wider and the face is shorter. And also histological and echographic analysis showed that these mice have eccentric cardiac hypertrophy. So in fact, we can kind of nicely recapitulate quite of some Noonan syndrome phenotypes. But what is about kind of cancer-related phenotypes? It's quite known that Noonan syndrome patients have a higher risk of getting cancer, but it was not the case in our mouse model. Even we were monitoring these mice up to quite old age, we couldn't observe any cancer-related phenotypes. So kind of we started to think maybe just still GAPLA insufficiency is not enough, and we switched to and tried different tissue-specific knockouts. So we started with the model in schwannomas, which are benign tumors of peripheral nervous system. These tumors are benign, but still they lead to quite devastating pain and quite some issues for these patients. And LZTR1 mutations were found in about 40% of familial schwannomatosis. So we generated Schwann-specific knockout of LZTR1. We were able to induce LZTR1 knockout around during embryonic development around 9.5. And again, we were monitoring this mice for up to 2 years, and we couldn't detect any schwannomas. So it was quite frustrating, and we went back to genetic data. And it's quite well described that the germline mutation of LZTR1 is also associated, first of all, with the loss of chromosome 22. And again, it includes the region where LZTR1 is located, so it's a full loss of function of LZTR1, but it's also lead to, quite commonly lead to the second mutation in NF2. And the NF2 is a known component of the HIPA pathway. So we suggested that maybe you need a cooperation between LZTR1 loss and NF2 loss or activation of the HIPA pathway to induce schwannomas. So, and that's why we generated a triple knockout mice where we have loss of LZTR1 and also LATS1 and LATS2. And at age about 1 year, we can now see that in fact there is a nice cooperation between LZTR1 loss and activation of the HIPA pathway, because we can quite big schwannomas through the whole body. So currently we are quite busy with characterizing this model and see what would be a potential mechanism that and a mechanism of cooperation between LZTR1 and the HIPA pathway. And the second model that kind of cancer-related model with generators is pancreatic specific knockout of LZTR1. Again, mutation, germline mutation of LZTR1 were recently published for some of patients with pancreatic cancer. So that's why we decided to generate LZTR1 flux model that with knockout of P48. In this case, we have LZTR1 knockout around E9.5. And what we can clearly see that even a single loss of LZTR1 could already lead to enlarged pancreas and quite some abnormal kind of canine-like structures, suggesting that in fact just loss of LZTR1 could predispose to pancreatic cancer development. And of course, for us, it was quite curious to see whether LZTR1 could also cooperate with oncogenic RAS. So if you are looking for pancreatic cancer, of course, it's quite known that pancreatic cancer is in 90% there are mutation of KRAS. And we also could see that the loss of LZTR1 is observed in about 30% of PDAC patients. So that's why we decided to model this. And again, we can quite clearly see that loss of LZTR1 is associated with much more dramatic stages of pancreatic cancer development. So again, we are getting these mice and we are trying to figure out what's going on and how LZTR1 loss could contribute to pancreatic cancer development. So, and the last model that we generated, we also look for lung cancer. And in case of lung adenocarcinoma, it's 30% of these patients are harboring KRAS mutation. And it's quite commonly occurring and quite significantly occurring with loss of heterozygosity of LZTR1, which is also observed in about 30% of lung adenocarcinoma patients. So, and for this, we decided to use a modified model of thyroid JAKs. So we combine KRAS G12D allele with LZTR1 flux and induce LZTR1 knockout and KRAS G12D expression by intratracheal injection of adenocree. But we couldn't use just a regular adenocree because in case of LZTR1 loss, it led to quite significant bleeding phenotypes. So that's why we decided to use CRE and SFTPC promoter that would specifically express only in alveolar type 2 cells. So, and when we started to monitor these mice, we could clearly see that LZTR1 loss led to early onset of tumors. In this case, we can already detect adenoma lesions about 5-6 weeks after injection. And then we monitor these mice by PET CT scan to measure volumes of these tumors. And we could clearly see that either loss of both alleles or just loss of one allele could dramatically facilitate the growth of these tumors. And we could also clearly see that loss of LZTR1 is associated with poor survival of these mice. And in case of lung cancer, we already have a clue what's going on. So we did proteome analysis of wild type and LZTR1 knockout tumors. And we could clearly see enrichment on proteins that are specifically expressed in immune cells and are specifically expressed in neutrophils. So this suggests that loss of LZTR1 could somehow modify the KRAS function and lead to modification of tumor microenvironment. So to check, one slide is missing. Sorry. Oh, somehow, yeah, one slide is missing. Sorry, but maybe we can. But we did FAX13 profiling of LZTR1 wild type and LZTR1 mutant tumors. And we found that, in fact, we could see enrichment of neutrophils specifically in LZTR1 knockout tumors. And we could also confirm it by immunostaining using like 6 marker that you can see that, in fact, LZTR1 loss is associated with increased neutrophil recruitment. And of course, the next question, whether this increased recruitment could be associated with this increased tumor initiation and increased tumor growth. We perform a neutrophil depletion experiment. So we were injecting anti-Ly6 antibody, and you can clearly see that we could get a nice neutrophil depletion. And while this depletion did not really affect the growth of LZTR1 wild-type tumors, it would significantly inhibit growth of LZTR1 knockout tumors. So this suggests that, in fact, this alteration in tumor environment that is triggered by LZTR1 is really important for tumor formation of specifically of LZTR1 deficient cells. So, and the question is, in which way LZTR1 can modify the activity of RAS oncogenes. So this data is still quite preliminary, but still what we can see is that if we again do pathway analysis of our proteome data, we could clearly see that loss of LZTR1 is associated with increased endocytosis and increase inflammatory response. And if we are looking for different branches of the RAS signaling pathway, of course, what you would think, first of all, it's that it might specifically increase the activity of the RAL TBK pathway. So we tested this hypothesis, and in fact, we could see that if we suppress LZTR1, this was done in cells with KRAS G12 mutation, we could clearly see that it leads to increased TBK1 phosphorylation, suggesting that in fact the LZTR1 loss is associated with increased TBK1 activity. And we already tried to see what could be effect of TBK1 inhibition on tumor growth. And again, we treated Amlex, we treated our mice with Amlexanos with a TBK1 inhibitor that actually quite cheaper than compound one that David is in favor. So that's why we use this one. And again, we can quite clearly see that while it's didn't really affect the growth of wild type tumors, it quite nicely suppress the growth of LZTR1 deficient tumors. So, and this is leading me to kind of a model that we have for a moment. So what I believe that if we are talking about disease that associated with wild type RAS, loss of LZTR1 function lead to dysregulation of RAS ubiquitination and could lead to upregulation of the MAP kinase pathway. This could be the case in case of germline mutation of LZTR1 associated with Noonan syndrome in schwannomatosis. It's a bit less clear about hepatocarcinoma and glioblastoma because we did some things in this direction as well, but I'm not sure how good this genetic evidence for LZTR1 mutation in this type of cancer. So it's still kind of under the question. And we could also see that the LZTR1 loss somehow could also modify the activity of oncogenic RAS. In this case, we don't really see a big change in activation of the MAP kinase pathway. That is quite obvious because it's already quite hyperactivated. But in case of oncogenic RAS, we could see quite a nice increase in the activation of RAL-TBK1 pathway. And we believe that by activating this specific branch, LZTR1 loss could modify the tumor microenvironment, and by this contribute to facilitation of cancer development. And just to summarize, I would like to thank my group who, of course, contribute to all these experiments, all my collaborators from Ghent who is helping me with all my spectrometry experiments, to NCBI group who did all in silico modeling for us, and for people who are helping us with all genetic characterization and providing all information of new data of LZTR1 mutations in patients, and of course to my antibodies. And thank you all for your attention.
Great talk. Great talk. Yeah, so definitely please, if anybody has any questions, please add them to the Q&A. And I definitely have some questions. And yeah, I'll forgive you for using Amlexinox. I get it. I'm just kidding. Yeah, I guess one big question I had is, so the speckles or the phase separation are very interesting. And I mean, do you think those are associating with the plasma membrane, or I know there's a literature about RAS signaling from endosomes. And so how do you sort of reconcile that? And do you think that some of the action is happening in endosomes?
Yeah, it's kind of, yeah, it's a difficult question. And one thing, yeah, it's still, I don't really know when this ubiquitination is going on. So yeah, I don't feel that there really this puncta, like organelles, I don't know how to call them correctly, that they're really associated with the membrane. But what we could clearly see, if especially when we overexpress in LZTR1, this puncta could definitely suck all RAS protein and dramatically inhibit RAS activity. This we can clearly see. But with you need a certain level also RAS expression for the formation of this puncta. This kind of we are trying to play around and try to see.
Sounds great. Okay, yeah, here's a question. It's in the chat. Actually, here we go. It's in the Q&A now. The question was about in KRAS G12D cell lines, have you tried sort of the in vitro experiment of knocking out LZTR1? I think you had shown in your prior paper about the MAP kinase enhanced signaling. But yeah, have you looked at G12D cell lines and looked at the effect on proliferation in vitro when you knock it down or out?
Yeah, we did actually, of course, before starting all these in vivo studies, especially, yeah, I have quite a high tendency to try different cell lines. We tried a lot. We tried different types of different cells, and actually what we could see, especially in case of KRAS mutant, or if like kind of more transformed cells, we do not really see any effect of LZTR1 loss on any, I would say, any of cancer-related phenotypes. We tried 2D, 3D growth, migration, whatever, in many of different cancer cell types. I could say we couldn't. I know that there is some publication, but in our hands we could see nothing. We could see only phenotypes, nice phenotypes, only in vivo.
It's maybe to follow up on that question, did you try any cytokine profiling of conditioned media to see if perhaps you're getting increased IL-6, IL-8, things that would recruit neutrophils?
Yes, for now, kind of the most significant one is it's not something that is really conventional. We have definitely increase in TNF alpha. We can see quite increase in VGF alpha. Alpha. For 6 no, it's kind of what I would expect. We did kind of a full secretome analysis, but then the difference are not so dramatic. So for now, it's TNF alpha, and I am a bit doubting whether it's something that people would be excited. It's kind of, I don't know, what do you think on this?
No. It's
kind of, but or CXL, no, unfortunately not. Mm-hmm. So, but you would really think that we should use your compound and try it?
Well, no, I think just
the price was really, I don't know, maybe it's something specific to Europe, but here it's the difference is really dramatic.
No, there have been multiple papers. Melexinox was first described as TBK1 inhibitor by Alan Saltiel for treatment of obesity in a Nature Medicine paper. But the concentrations that are used are very high, and in terms of, I mean, it probably does inhibit TBK1 activity. So the experiment's correct. It's just in terms of a kind of a pharma compound. There are definitely many companies that have potent specific ones, and that's one that. But yeah, the challenge is it's expensive. But for proof of principle, it's very interesting. So. Let me see. I think here's another question. Yeah, do you think that LZTR1 is the only enzyme to monoubiquitinate KRAS? And also, what are there specific lysine residues? I know you talked about early on about residues that you focused on, but I don't know if we heard too much about. I think you mentioned it in one early with HRAS, but yeah, the specific residue on KRAS that it might be regulated.
So yeah, I mentioned because there are also other publications. So we are mostly working on 128 and 170. It's also could be ubiquitination on 147. But the problem with this one, it's located in the GTP binding pocket. And then what I don't like, what other people are doing, because if this pocket is really conservative, and if you're trying to touch it with any mutation, it would affect the conformation of the pocket. And of course, then GTP is less bound, and GTP is less bound, and you have higher concentration of GTP, and then of course it would immediately lead to activation of the protein. So it's always a problem that I'm trying not to touch. There is also 1,4 that is also could be acetylated and ubiquitinated. For this, it's shown that it also could suppress activity. In case of different ubiquitin ligase, so several were published. One is Rabix5. For this, I don't trust it. It's called also RAB-GEF1. It has a quite nice ubiquitin binding domain, and I don't think that this one even it was a publication from the Barsagi group. I don't know. I put quite some efforts. I couldn't reproduce it. There are also several other ones that are published, but like NET4, TRCP, beta, but this, I never really checked myself, so it's like, but in the literature, yes, it's known that several others could contribute to RAS ubiquitination.
Great. Well, I know we're hitting the 5 o'clock.
None of them are really so nicely associated with RAS-related disease as LZTR1.
It's robust. Well, I wish we could give you a round of applause. It's hard to do that on Zoom, but yeah, it was a really amazing talk. And again, we hope to see you in person Welcome back virtually to Dana-Farber, and thanks again for an awesome talk.
But yeah, if you have any suggestion or whatever, if you can write me a note, yeah, I would really appreciate your feedback. Because the idea is to really see if to kind of use LZTR1 loss as a kind of kind of a marker that it could be more efficient for TBK1.
Yeah, we should set up a Zoom. Yeah, we can set up a Zoom.
Okay.
Take care and great job.
When you have time, I will be happy to discuss in more details.
All right, great to see you, and very well done.